Stem cell regenerative therapy is one of the newest technologies available to aid in the restoration of damaged tissue or organs. Not much research has been done to explain the exact mechanism as to how it works, but the positive response horses have to this treatment is undeniable. I am currently working on formulating a research project with Dr. Johnson investigating the differentiation capacity of certain stem cells in horses. Stem cells are immature cells that can develop into any type of cell to assist in the healing process of damaged tissue. The FDA defines cell therapy as the prevention, treatment, cure, or mitigation of disease or injuries in humans by the administration of autologous, allogenic, or xenogenic cells that have been manipulated or altered ex vivo. When an animal is injured, stem cells naturally self-renew during the healing process. Stem cell therapy aims to speed up this process by amplifying the number of stem cells through culture ex vivo and injecting them back into the horse. Stem cells are derived from three main sources in the horse, including the bone marrow, adipose tissue, and the umbilical cord. Currently, stem cells as equine repair aids are limited to adipose-derived and bone marrow-derived mesenchymal cells. Bone marrow mesenchymal cells tend to be better at conversion than adipose mesenchymal cells, suggesting that they are more plastic in nature. Both types, however, are a mixed population, which may be the reason that there is very little engraftment and mixed results following injection into the tendon. The cord tissue contains the Wharton's jelly of the umbilical cord, which will be my area of research. Bone marrow extraction of stem cells is the most studied method of the three. Commonly, in adults, the cells are taken from the sternum. For horses under 5 years of age, the cells are taken from the hip. Some disadvantages to aspiration of stem cells from bone marrow are that the potency and number of cells declines as the horse ages. The max number and potency is achieved with horses under 18 months of age. The procedure is also very painful and invasive, and there are not many mesenchymal cells in each sample. Adipose tissue contains a large number of stromal cells. Because it is easy to obtain in large quantities, adipose tissue has been found to be an ideal source of uncultured stromal stem cells. Adipose tissue, like bone marrow, is derived from the mesenchyme and contains a supportive stroma that is easily isolated. Being abundant, accessible, and replenishable, adipose tissue is an attractive source for adult stem cells that can be isolated by collagenase digestion and differential centrifugation. However, this technique is invasive and can prove problematic in young and fit racehorses without an adequate amount of fat to be collected. This figure shows the anatomy of the umbilical cord. The umbilical cord contains 2 arteries and 1 vein surrounded by the Wharton's jelly, which insulates and protects the umbilical cord vessels. Umbilical cord blood proves an excellent source of stem cells, not only because of the ease of collection, but also for the probable higher proliferative potential of the stem cells and increased flexibility for differentiation. However, cord blood provides lower overall numbers of mesenchymal cells than bone marrow, cord tissue, and fat. My research involves using Wharton's jelly or umbilical cord tissue as the source of stem cells. Wharton's jelly-derived mesenchymal stem cells have been shown to have the capacity not only to differentiate into adipogenic, osteogenic, and chondrogenic cells, but also into neuronal, endothelial, cardiogenic lineages. The higher proliferation ability, differentiation capacity, and non-invasive extraction procedure of stem cells from the umbilical cord tissue are just a few of the reasons we decided to use Wharton's jelly as our extraction method of choice. VetStem, located in Poway, California, is a current leader in stem cell therapy to help injured horses recover from soft tissue damage. Current research is also being done at the University of California, Davis, and at VetCell in Cambridge, England. VetCell derives stem cells from bone marrow and has used its therapy on approximately 1,700 horses so far. In a study of 170 jumping horses monitored through treatment and rehabilitation, Researchers found that about 80% of them could return to racing, compared with previously published data showing that about 30% of horses given traditional therapies could return to racing. Additionally, after 3 years, the re-injury rate was much lower in stem cell treated animals. The overall process of stem cell therapy begins by harvesting the cells from the horse in question, which in this diagram is from the bone marrow. The tissue is then sent to the lab where stem cells are selected and multiplied. They are then returned to the vet who injects them back into site of tendon injury, using ultrasound-guided implementation. These stem cells will develop into tendon cells to heal the injury with the correct type of tissue rather than forming scar tissue, which is what would regularly result without stem cell therapy. One case study performed at Root and Riddle Stem Cell Lab used stem cells derived from the umbilical cord of newborn foals. These are radiographic images of a 10-year-old thoroughbred mare with severe sinking of both front feet before and after stem cell therapy. The mare was treated with deep digital flexor tenotomy foot casts and partial wall resections. The foot did not show any signs of improvement for over 2 months. Stem cell therapy was begun at this time along with foot casts and antibiotics. 2 weeks later, the coronary band and sole began to show healthy growth. The after radiographs on the right show the foot several months after stem cell therapy. Be sure to note the thick sole depth and fairly healthy appearing coffin bone. These ultrasound images are from a vet cell lab and show the tendon in a horse's front leg. An area of damage can be seen in the yellow circle in the top picture. Looking at the bottom image, you can see the area has healed after the injection of stem cells derived from the animal's fat tissue. Stem cell therapy usually runs around $2,000 to $3,000, plus the cost of diagnostic work, ultrasound, and consulting. Therapy is most commonly utilized in the racing and showing industries because of the associated expense. For my research with Dr. Johnson, we chose to use cord tissue because obtaining cord blood is labor-intensive. In the past, Dr. Johnson has failed to collect from several mares because she either missed their foaling or the cord broke before she was able to collect blood. Maiden mares are especially prone to standing up before collection is possible. Umbilical cord mesenchymal stem cells may also offer an advantage in that they are more immunotolerant, which means that you don't need to use a horse's own cells for injection, but can use umbilical cord mesenchymal cells from one animal into another. This means they are allogenic. Currently, since no species-specific antibodies exist to characterize equine stem cells, my project is to research a subset of cells marked CD133. Like the bone marrow and adipose mesenchymal stem cells, umbilical cord cells are mixed, which means I must isolate specifically for CD133. Presently, vets inject all of the mixed population of cells back into the animal after proliferation. So by isolating CD133 specific stem cells through my research, we will be looking at solely the differentiation capacity of the stem cells. CD133, also called prominence, is a surface glycoprotein that denotes a stem-like cell. These cells may be the magic bullet for injection into tendon lesions. I will isolate the mixed umbilical cord mesenchymal cells and the CD133 subgroup and test their ability to differentiate. We will include control-positive reacting antibodies that include CD90 and CD29 markers that are commonly used in adipose and bone marrow mesenchymal cells. Both antibodies recognize components of the integrin system, which are found on multiple cell types and not restricted to mesenchymal cells. Negative controls will include antibodies against blood cells CD34.